Alright, now let's dig deeper and talk about phloem. The phloem, like what I've said from our previous slide, it moves food substances that the plant has produced by photosynthesis to where they are needed for processes such as growing parts of the plant for immediate use, including the flowers, the shoots, or even the stem. Second one for storage organs such as the bulbs and tubers, which are present in potato, in banana, and other dicot plants that we have here. And lastly, for the seeds that are developing, about to emerge into becoming a plant or a flower. That is the main function of phloem. It transports food molecules from one plant organ to another plant organ. Okay. Transport in the phloem therefore takes place both up and down the stem, which is very contrary to transport in the xylem, which is just upwards. Take note of this. Transport in phloem is very versatile because it happens up and down, unlike the xylem, which is only upwards. Lastly, I would like to mention that the transport of substances in the phloem is what we call translocation. Hence, as the name implies, location, it has the ability to transport or move plant molecules from one organ to other parts of the plant, including the stem, the leaves, the bulbs, the root tip, or even the flowering portion of a particular plant. For this image, you will see the cross-section of a phloem in a root. Phloem are comprised of living cells, namely sieve tubes and companion cells. The cells that make up the phloem are adaptive to their changes or function. Let's discuss the former, which is the sieve tube or sieve plate, which is located on both the medial and the lower portion of the phloem. They are specialized for transport, and I would like to add up that they have no nuclei. Each tube has a perforated end, so its cytoplasm connects one cell to the next. Again, the main function of cytoplasm is that this is where the sieve plates are suspended to. Basically, it is a fluid cavity wherein it also allows for the supporting of the sieve plate to companion cell, which is located just underneath the sieve plate or sieve tubes. Basically, this is how these plates look like. They are hexagonal in shape with a lot of plates inside it. And as for the companion cells, transport of substances in the phloem requires energy. Not only a liberal amount of energy, but I am referring to an ample amount of energy. When we say ample, that indicates that it requires a lot of energy, wherein one or more companion cells attached to each sieve tube provide this energy. This is where the companion cells are located. As you can see, it has a nucleus that regulates the various cell activities that takes place in the phloem. Lastly, a sieve tube is completely dependent on its companion cells, as the word implies companion, responsible for having control in the different cellular activities that can take place inside a root, specifically in the phloem region. I would like to mention everyone that on this cross section, you can see that we have two tubes of phloem running parallel from each other. Next slide, please. Let's discuss the comparison of transport in xylem and phloem. For the type of transport, in xylem, it undergoes physical process. And for the phloem, it requires energy. As for the substances transported, we have water and minerals for xylem, and for the phloem, products of photosynthesis, which includes sugars and amino acids dissolve in water, making these sugars hydrophilic, which means that they can be blended or mixed with water. They are water loving. For the direction of transport, like what I've said a while ago, xylem undergoes upwards movement or transport, and as for the phloem, they can be both upwards and downwards. Next slide, another cross section through a plant root. The difference between phloem and xylem. The xylem and phloem are distributed differently in roots and stems. In the roots, the xylem forms a central column, which is these spherical series that you are seeing right now. This is exemplified by this cross section that you have on your screen right now. And again, this is the central column that we have here, wherein the yellow structure that you are seeing is scattered on each xylem is what we call the phloem. It forms a solid support. The phloem is towards the center, outside the xylem. However, in the stem, the transport tissues of the xylem and phloem are grouped into vascular bundles, which is a series of spherical shaped structure that are running towards the center of both xylem and the phloem. For the first photo, this is the plant root, and as for the second photo, this is the stem of a plant, wherein it is very prominent that the presence of phloem and xylem are visible here. Okay. Proceeding to our next slide, let's talk about the major differences between source versus sink. Source versus sink. The former is the part of a plant where substances are produced. These parts include the leaves for sucrose, amino acids, or enter the plant. And as for the latter, which is the sink, it refers to the part of the plant where the substrate can be stored. In the case of roots or stem for starch. I hope that is clear. Again, as the word implies, source, this is the source where sugars are produced. And as for the sink, this serves as the storage for the substances produced by the source. Examples, it can be categorized into sources and sinks. For the sources, it can be found in both leaves and root hairs. For the former, sucrose is produced here. Again, sucrose is a form of sugar that is present in most plants. And as for the root hairs, nitrates are absorbed here. What about the sinks? For the roots and stems, starch is stored here for quite some time until it undergoes photosynthesis. And for the root tips, amino acids are produced here, which is exemplified for this model that you have here. Again, for the roots or sa ugat, it functions many different responsibilities. This is where the synthesis of proteins takes place, absorption of water minerals, which enables it to transfer to other organs of the plant. And of course, there is the storage of starch, which can be utilized for photosynthesis. And for the transport system, this is where the water transport system and nutrient transport system occurs. Protein, fat, and starch are stored in some portion of the leaves as well as evaporation of water. At the upper portion of the plants where the leaves are connected to internodes. This is where the synthesis hormones of proteins take place, as well as photosynthesis of sugar and other amino acids occurs. This is the illustration between the various portion or regions wherein source and sink are involved. In addition to that, when a plant is actively photosynthesizing and growing, the leaves are generally the major sources of translocated materials. So when we say translocated materials, this can also be called as assimilates. Hence, the topic that we're having, photoassimilates, which includes the involvement of light to the various end products of photosynthesis. Again, translocated materials can also be referred to as photoassimilates or assimilates for short. They are constantly producing or generating sucrose, which is carried in the phloem to all other parts of the plant. These parts, which is specifically the sinks, includes the roots, the flowers, and the fruits, of course. Let's talk about the roots first. It may change some of the sucrose to starch and store it. And for the flowers, it utilizes the sucrose to make fructose. Basically, it's all about converting a sucrose to another type of sugar in the form of fructose. Later on, when the fruits are developing or on the verge of ripening, quite large amounts of sucrose may be used to produce sweet, juicy fruit ready to attract animals. Not only that, but we are all quite familiar with the succulent fruits that we have here, those that are citrusy, which can also be utilized to be consumed by the different animals, specifically those that are pollinators, including the bees and butterflies. Okay, for this image that you are seeing right now, this is the general step or process of the source versus sink. For the first step, sugar is actively transported into the sieve tube members of the phloem. Water flows via osmosis, and this is the source cell that we have here. Apparently, they are scattered everywhere in the sieve tube from the top region to medial region up to the lower portion of it. By which, having said that, sink cells are scattered on this structure that you are seeing right now in a radish, which is the model of fruit that we are utilizing. Second step, higher water pressure at the source forces the phloem sap to move toward the sink. So when you encounter the word sap, this is the water that has been produced in the sieve tube, by which it's been converted into the form of sap. Lastly, sugar is unloaded at the sink and the water returns to the source via the xylem. The process is interchangeable because it could be the other way around. The presence of source cell can also be present in the xylem structure that we have here and vice versa. Okay, so that's it for this image that we have here. Now, for additional information regarding source and sink, many plants have a time of year when they become dormant. When we say dormant, that means inactive, not performing any activity or cellular processes. And during this stage, they wait out harsh conditions in a state of reduced activity. Again, making them stagnant or making no progress at all. Dormant plants, on the other hand, they don't undergo photosynthesis or they do not photosynthesize, but they can survive on their stored starch, oils, and other materials that give them sufficient supply of energy. When the seasons change, they begin to grow again. Now, The stored materials are then converted to sucrose and transported to the following region. For instance, we have a potato plant that is not able to survive the cold frost of winter. And during the summer, by which it is on the peak of having a super high temperature, the leaves photosynthesize and send sucrose down into underground stems. And here, swellings called tubers developed. The cells in the root tubers change the sucrose to starch and store it. What I'm trying to insinuate here is that potato plants really thrive during the summer season because there is a sufficient amount of sunlight that can lead to the process of photosynthesis, allowing the production of starch. However, in autumn or fall, the leaves eventually die. Through the process of senescence, nothing is left of the potato plant above ground, just the stem tubers beneath the soil. Lastly, in spring, they begin to grow once again with new shoots and leaves. The starch in the tubers is changed back to the sucrose and transported in the phloem to the growing stems and leaves. Lastly, it will continue until the leaves are above ground and photosynthesize and go back to different seasons once again from summer, autumn, spring, with exception to winter. Like what I've said a while ago, potato plants do not thrive in the cold condition or climate. Now, let's discuss a brief overview about carbon allocation and partitioning. Carbon allocation, as what's been written on your screen right now, is a critical physiological process where the products of photosynthesis are shifted between respiration and biomass production, ephemeral and long-lived tissues, and above-ground and below-ground components, wherein changes in color allocation significantly affect the growth of individual plants and terrestrial biogeochemistry via influences on litter quality and decomposition rates, carbon and nitrogen sequestration, and plant atmosphere gas exchange. When you encounter the term nitrogen sequestration from the verb sequester, it means that it excretes or gets rid of nitrogen from a plant. So basically, sequester is the removal of waste products from a particular plant. Lastly, I would like to point out that theoretically, plants preferentially allocate carbon to acquire the resource that most limits their growth, which is one of the most important mitigation processes for reducing the impacts of unfavorable environments. Now, for our last slide, what about carbon partitioning? Carbon partitioning is the process whereby assimilates or the end products of plants using photosynthesis are distributed throughout the plant body from photosynthetic tissues. For most plants, this occurs by loading suck into the phloem and transporting it from source tissues in the form of net exporters to sink tissues, or what we call the net importers, where suck is unloaded. Lastly, this process is well characterized at the physiological, biochemical, and anatomical levels. So, I guess that's it for our pre-lab discussion about photoassimilate transport. And now, let's proceed to your lab activity that you will be conducting within the week. So, I will share it to you. I'm still thinking if I should do it individually or by group kasi you'll be having a reporting again next week. So itong ating lab exercise number 4, the photo assimilate transport. You can see the introduction as well as the objectives and materials here. And for the procedure, you are required or tasked to get a specific portion of the stem of a plant. You have to mark the area where you will cut or remove a ring on the plant's stem. Note that it should be below all the leaves. Second one is to measure the girth of the die-cut system, both top and bottom on the ring mark. And this will be your initial data between the centimeter of the girth of the die-cut stem once again. Using your preferred cutting tool, it could be a scissor or a knife or anything that you'd like to use to be able to make a cut in the stem. Remove a ring of bark along the stem of the die-cut plant. And you have to expose the plant to the sunlight to let it photosynthesize for a day or two or even more if that is what you prefer. Also, water the plant accordingly. Okay. Next one, you have to monitor the plant with 6-hour intervals, daytime only, not nighttime, and measure the girth to know if there are changes in size. Lastly, tabulate your results and include pictures of each datum. This is the table wherein you're going to put your result. We have the 0 hour. So for this hour, this is where you're going to measure the girth of the die-cut stem the moment you obtained or cut the stem. So that would be your initial data, followed by 3rd hour, 6th, 9th, up to 24 hours. And this can be applied to the above the ring mark and followed by below the ring mark. Same process as the first one. We have the 0 to 24 hours or more if you would like. Don't forget to answer your observation with discussion and analysis. And here are the guide questions that you must be able to answer. How is the structure of phloem sieve tubes related to its function? Why can't girdling or ringing experiments be performed in monocots? Third, are the functions of source and sink interchangeable? Pwede ba silang pagpalitin? Why or why not? Number 4, explain the relationship between light intensity and translocation. And for this particular portion, you are tasked to illustrate the direction of flow of photoassimilate transport. It should contain xylem, phloem, sink, and source. Explain the process. You can do it manually or digitally. It's up to you, however you'd like to do it. And explain the effect of the following factors to the regulation of phloem transport. We have the growth hormones, mineral deficiency, light, temperature, microbes, carbon dioxide, water, and salt stresses. Okay. If you have references, do not forget to put it here on this portion in APA format para ma-acknowledge naman sila. And here are the grade rubrics that you must be able to comply with. O sige, by group na siya, everyone. Oo, kasi I think parang mahirap pag individual. So I've decided to do it grouply. You know who your leaders are, okay? And just like the usual, you have to put the leader here, the members, student number, and don't forget this one, the details of participation, because I am grading you according to your contribution from 1 to 10. 10 being the highest grade. So make sure that you will be able to coordinate with your group members to come up with a decent work. And for this one, affix your e-signature as well as your name, which confirms that you have been able to contribute or to chip in this activity. So, ayun lamang po. By group siya. Sige. We'll send it later sa ating classroom. Tapos sa reporting, once again, you will do it next meeting. So, prepare nyo na po yung inyong PPT slide para ma-present yung data or observation nyo po. Okay. Do you have questions so far? Or is everything clear? Any concerns that you'd like to raise? Anyone? Or clear tayo sa activity? Questions? All goods naman. How about the others? Okay. But still, kung may katanungan regarding sa activity, paki-chat na lang po ako sa FB or paki-mention yung aking name sa ating chat group so that I will be notified that you have sent me a private message. Are you guys sure? No more questions? Okay. So, clear naman. If that is the case, again, BS Bio 4-5, thank you so much for your presence. I appreciate you all. May you have a blessed day ahead of you. That ends our synchronous session. God bless you all. This has been Sir Rai. See you guys next week for your reporting. Thank you very much. Bye everyone. Class is now adjourned. Thank you. Bye.